Okay, so we left off with polygenic. So if you recall, polygenic basically is a situation where you have one trait that's governed by multiple genes, right? And so the more genes you have, the more variables you have controlling that trait, which basically means instead of having categorical data like column A, column B, column C, and nothing in between, the more data you have, the more points you have in between those columns, right? So instead of having A, B, and C, you have like A.1, A.2, A.3, right? So kind of like height, right? Height is a perfect example. Right? You don't just have like 4 foot people, 5 foot people, 6 foot people. You've got like 4, 4.1, 4.2, 4.3, 4.3.5, 4.3 3/4, right? I mean, so you have every potential data point in between there. That's called continuous. So the more data points you have, that is to say, the more variables you have, which in this case is genes, then the more continuous you are, right? And that's kind of what creates this sort of continuous distribution. So height is an example. Skin color is another example. I think skin color is controlled by something like 8 genes. And the alleles, there's 2 alleles per gene, but the alleles are basically very straightforward, right? So it's either you've got dark or light, right? That's kind of your 2 options in terms of pigmentation. And then what creates the pigmentation is just the number of dark or light alleles that you inherit, right? So, and generally speaking, since you have multiple genes involved in it, and actually it's not just genes in this case, right? Their variables are actually the number of alleles. So if you have 1 gene with 2 alleles, you're going to have more dark pigment than if you had 1 gene of dark in there. So when you have all these 8 alleles, then what you see is kind of a bell curve, right? So basically you have a very, very small segment of the population that has all light alleles. So seeing somebody who's very, very pale is very rare. And then on the other side of things, if you have all dark alleles, seeing somebody who's very, very dark is also very rare. The majority of the world population is somewhere in the middle. Mixtures of light and dark alleles, but basically you kind of have this sort of median sort of olivish tone, right? So you're either kind of like an olive leaning dark or olive leaning light, but you're kind of in that region. So the big bolus of the bell curve is somewhere in the middle. And so that's a polygenic trait. The next one, which is still a violation of Mendel, the first criteria of Mendel, right? 1 gene, 1 trait, is pleiotropy. So when we take a look at pleiotropy is a situation where you have 1 gene which controls many traits. Now, generally speaking, with pleiotropy, it's usually a disease gene is what you're looking at. And the example that we have here is a couple of them. Sickle cell anemia, which is all right, but it's not as variable. I like cystic fibrosis better because cystic fibrosis is a very, very difficult thing to deal with. And it's very simple, genetically speaking. All it is a mutation in a chloride channel. That's it. So remember, we're talking about facilitated diffusion. You had sodium channels and potassium channels. This one happens to be a chloride channel. It's a mutation chloride channel. So you have a fundamental inability to be able to move and transport chloride normally into and out of the cell. So that's just 1 gene. However, when you take a look at the symptoms of cystic fibrosis, you have lots of symptoms. And each symptom set is considered a specific or different phenotype, right? So if you take a look at cystic fibrosis, you're going to see some symptoms that are associated with the liver, right? So fat digestion, and so these kind of are all associated with different symptoms in the liver. Pancreas, you also have digestion, vitamin absorption, that nature. So that's a different set of symptoms. That would be considered a different phenotype. You have a respiratory thing, right? So your cilia don't operate normally. So your cilia don't have the ability to actually sweep the mucus out of your respiratory tract the way they're normally supposed to do, right? So normally cilia in your respiratory tract, mucus up out of your respiratory tract because the cells that they're attached to secrete mucus, which traps particles in the mucus so you don't inhale those particles. So it's kind of like an air cleaner, essentially, if you will. But the idea is the cilia are designed to sort of sweep this sort of particle-laden mucus up out of your respiratory tract into your cough reflex so that you can cough it out. Well, the problem is if your cilia aren't able to flush all of this stuff out, then what happens is it stays inside your respiratory tract. And then by and large, it's probably going to go down, right? Because it's not going to hit your cough reflex unless the cilia sweep it up there, okay? And then, of course, that's trouble, right? So if it starts going down into lungs, then you don't want stuff going into your lungs. If it gets excessive, yeah, I mean, you would have to kind of like get it out of there. You have to like suck it out of there. So it's not pleasant. But that would be a totally different set of traits. Right? That would be a different phenotype. And then, of course, but you're expelling excess chloride. Why? Because you're not processing it normally. So you are treating it like a waste product. You have instead of moving it to where it needs to be, it's accumulating it sort of waste, and you have to get rid of it. We have excessive chloride. So all of these are considered separate phenotypes. It's all caused by one gene. Listen. It's the digestive one, because when you're doing your digestive buffers, you may recall this because you've had AMP2, but when you're actually secreting the acid in that process, you have to use the chloride shift in order to balance out your charges, and that's where you fail, is in that chloride shift part of it. So it has nothing to do with insulin, because chloride's doesn't have any role to play in the endocrine portion of the pancreas. So the next one is going to be multi-allelism. So now what we're doing is we're messing with criteria B. What happens if you have multi-allelism? What is this? This is basically where a gene has more than 2 alleles in it in the population. Notice I didn't say in the individual, in the population. You only have 2, right? Because you only have 2 parents. There's no such thing as having 3 parents. You only have 2, right? So you're only going to ever have 2 of these, but the population at large has 3 or more. Right? There's some genes that actually have quite a few alleles in them. So multi-allelism is a violation of the allele situation. But what I want to do is, and the best example of multi-allelism is the ABO blood grouping. And we'll talk a little bit about that. But what I want to do is I want to put this one on hold, because the ABO blood scenarios actually also are example for codominance, which is a violation of the next one, the 3rd criteria of mental. So what I want to do is I want to finish out the violation of the dominance situation first. We'll talk a little bit about incomplete, and then we'll kind of circle back around. We'll pull multi-allelism codominance together as we talk about the ABO blood group. Okay. So let's take a look then at violations of number 3, that is to say complete dominance. We have 2 different versions here. So you have what's called incomplete dominance. This is where the dominant allele doesn't fully mask or completely mask the recessive. We'll take a look at that. We've actually already seen that, so we'll kind of point that out and sort of fill that idea in. Then you also have what's called codominance. And codominance, this is basically where 2 dominant alleles express equally. So they're equal parts. One is not overshadowing the other, right? There's none of that. So incomplete dominance, we've actually seen, right? Because remember, we talked a little bit about this earlier. We started the chapter off talking about what? Crossing red flowers to white flowers, and what you get? Pink, right? So that's an example of codominance. The reason why it's pink is because the red dominant is not strong enough to completely overshadow the white. So what you're seeing in pink is not just a kind of a bleaching out of the red, but what you're seeing is a bleeding through of the white. Okay. So that's because the red is not able to completely overshadow that recessive. And then of course, codominance we'll talk about with the ABO blood group. But what I'm going to do is I'm going to show you first of all an incomplete dominance example. And in this case, what we're going to be taking a look at is 4 o'clock plan. So if you take a look at this, you'll notice there's a couple of little bit of a notation situation that I want to sort of clear up. So generally speaking, in genetics, there's multiple ways to sort of do your notation, right? So for the most part, the most common one that we see is the uppercase letter, lowercase letter for dominance and recessiveness, right? So that's kind of the most common one. That's also the one that's associated mostly with sort of introductory and lower level genetics. So when you're like in an introductory course, that's probably where you're going to live. If you're looking at like a genetics lab, then oftentimes you'll use alternative notation systems. Like for instance, if you're in human genetics, sometimes you use kind of a notation system that's a little bit different than, say, for instance, somebody who's working with flies, which has a slightly different notation system than somebody who's working in corn. And so you have different systems. And the reason you have different notation systems, not to be complicated, but because you're trying to basically sort of organize information that you have, and each organism is slightly different than the other. And so you have to have a slightly different organizational system to kind of keep track of things. This is a pretty common one that we see here, where what you start off with is a stock variable. So in this case, capital letter C, and that C stands for the actual phenotype. In this case, we're looking at color. So that's the abbreviation for the phenotype itself. And then in the superscript, you actually write the nature of the allele. Right. So for instance, in this case, superscript R is the dominant for red. Superscript W is the recessive for white. No, it's always right. So in this case, if you take a look at this, this is basically our cross with 4 o'clock plants. We take a true breeder for red. We can see that, right? Because it's got 2 dominant red alleles. Cross it to a true breeder for white because it's got 2 white recessive alleles. And then what do we get? We get the heterozygote, right? Which is pink. Familiar? That's what we started off with, right? When we talked about blender. It's not blending. It also should look familiar, right? At this particular point, are we doing anything different than what Mendel did? No. Our results are different, yes, but what we're actually doing is not. So let's keep doing what Mendel did. Let's take our 2 F1s, our 2 heterozygotes, and cross them. 2 pinks and two pink, right? Pink to pink. And then we're going to get the same Punnett square, right? So you basically get 1 red gamete and 1 white gamete. Those are the possibilities from each parent. So 1 red and 1 white. Okay. And then when you pair these guys together, then you're going to get a red and red, right? If you pair these 2 guys together, you're going to get 2 heterozygotes. And then the 2 recessives paired together are going to be 2 whites, right? 2 recessives. So that's your recessive. Now let's take a look at this, right? Notice what I circled. I circled the genotypes, yes? So what do I see? A ratio that is 1 to 2 to 1, right? So that's my genotypic ratio. I have 1 homozygous dominant, 2 heterozygous, 1 homozygous recessive. So my genotypic ratio is 1 to 2 to 1. So far, does it look like it's any different than Mendel? Mendel also saw a 1 to 2 to 1 genotypic ratio, didn't he? Okay, so wait. Now let's take a look at the phenotypic. So what do I have then with the phenotypic? Well, I have 1 red, 2 pink, and 1 white. So what's my phenotypic ratio? One to 2 to 1, right? 1 red, 2 pink, 1 white. So this actually is both phenotypic and genotypic ratio. So that's where it kind of it diverges from what Mendel saw, right? What was Mendel's ratio? What did he see phenotypically? He saw 3 to 1, right? With a 1 to 2 to 1 genotypic. In incomplete dominant, you see a 1 to 2 to 1 for both. Now, this is important, right? Because this is like when we look at these sorts of patterns of inheritance, these rules by which genes segregate, the way that we discern that there's a new set of rules at play is by these ratios. What we do is we essentially do the exact same thing that Mendel did, either a monohybrid cross or a dihybrid cross. And instead of getting his ratio, we get a different ratio like this, 1 to 2 to 1. As we study it, then what we realize is like, well, wait a minute. The reason why we have a different ratio is because we have a different set of rules here. And so not only that, but this ratio is diagnostic for the set of rules. So if you give me some random plant that I've never seen before and a random trait, and I do exactly what Mendel did, and I see a 1 to 2 to 1 phenotypic ratio, then I can say with confidence that this is incomplete dominance is what we're seeing, right? So that's the way we use the ratios. And that's what we did in these extensions of Mendel. It's kind of what we do in the genetics class as well. We kind of take a look at these alternative ratios and we use them to say like, okay, so this is the ratio for this, and this is the ratio for this. So this is different sets of rules. And then we kind of get into those sets of rules and try to figure out, well, what are these rules, right? How does this differentiate? So we got a question. No, incomplete dominance is the same in snapdragons as it is in 4-o'clock plants. So the rules are ubiquitous across all taxa. Okay, now let's go ahead and circle back around and pick up our codominance and pair it up with multi-allelism and put it together with our ABO blood grouping. First of all, what are the multiple alleles? Well, in the ABO blood grouping, we use the exact same notation system, right? So our stock variable is going to be capital I, which actually stands for immunoglobulin. And in this case, our alleles are either going to be big A, which is a dominant allele, big B, which is also a dominant allele, or little I, which is a recessive allele. So notice in the population, you have 3 different alleles. So that's multi-allelism. Now, when you have A and B together, they are codominant. So they're dominant to I, but codominant to each other. That's what this is saying. Okay. So they behave as a true dominant. They're not an incomplete dominant because when you put them with the recessive, you don't see the recessive at all. So let's take a look at this. Here's our blood types. And if you recall from the lab, our blood types basically are a glycoprotein set where you have different forms of a particular carbohydrate on each cell. Okay, so for instance, in the A-type person, the carbohydrate that they're making is galactosamine. Now, when an A person is making galactosamine, their immune system is growing up with that galactosamine. So their immune system is being trained to recognize that galactosamine is normal. And it's also being trained to kill all else. Now, a person with B type is going to be making the galactose. That's going to be their carbohydrate. Same thing's happening with their immune system. So their immune system is being trained to see galactose is normal, so everything else is killed, right? Now, an AB person is going to be making both galactosamine and galactose. And then an O-type person makes nothing. Okay. So if you then take a look at what your immune system is doing, what types of antibodies does an A-type person make? Against B, right? Because B is what it doesn't know, right? So it makes antibodies against what it doesn't know, anti-B. A B-type person makes antibodies for what? Yeah, against A, right? Because A is the foreigner in this case. An AB person, none, because it knows both, right? Its immune system is like, well, I see A and B, so I'm good with this. An O-type person, yeah, so it's making both of them, right? So everything looks crazy to an O-type person. So these are your antibodies that you're basically making. Now, the important thing to also understand is that these guys right here, this is your phenotype. So when you take a look at your blood type, it's what I refer to as your physiological phenotype. This one here is your molecular phenotype. Now, oftentimes students kind of, it takes a little bit of a process to sort of wrap their heads around that one, because oftentimes we're trained to think phenotype is something that's visible, right? We look in the mirror, there's our phenotype. we're oftentimes very focused on the visible. So light hair, dark hair, blue eyes, dark eyes, right? That's kind of what we're focused on. That is a phenotype, but your phenotype, remember, is really not necessarily how you look, but your phenotype is any physical manifestation of a gene as it's being expressed, including your molecules. So every time your gene makes a protein, that would be considered a molecular phenotype. Okay. And then, of course, blood type, because it's a physiological sort of a thing, that would be a physiological phenotype. So you can see the phenotype can actually exist on multiple levels. And it depends on the way you look at it. Now let's take a look at the genetics. In terms of the genotype, which is this guy over here, the genotype of a person with A-type blood can be one of two things, right? So you can either be homozygous dominant, which is this guy right here, or you can be heterozygous, which is this guy right here. So you can either be big A, or big A, little I. Either one of those will be A-type blood. Same thing with B. You can either be homozygous dominant or you can be heterozygous. That's going to give you B-type blood. Notice that the A and the B are fully dominant to I. Now, in an AB person, you're going to have one A and one B. But notice I actually was careful to draw as many A's as B's over here in my red blood cell, because I wanted to illustrate equal expression. So A doesn't have more, B doesn't have less. There's not somebody has the upper hand, the other one has the lower hand. There's not a Batman and a Robin, right? So the cool guy and the guy that everybody just ignores. Sorry. Robin, yeah, I'm not a Robin fan, sorry. Mostly because Batman did most of the time he was trying to rescue Robin. So Robin was more of a liability, actually, if you think about it. I'm just saying, if you're a Robin fan, great, he needs friends too, but just not coming from me. But so it's equal. And then so that's the example of codominance. And then of course your recessive has to have two recessive alleles. Yeah, so that's your homozygous recessive. Now when you take a look at your blood, the consequences of that, then when you take a look at donation, and this would actually be kind of an extension of your physiological phenotype, right? So basically this your reality essentially of how you operate. So when you take a look at an A-type person, can they take blood from a B-type person? No, because it's foreign to them, right? They'll attack it. Can they take it from an AB person? No, because the B portion of the AB person will trip their immune system, right? Can they take it from an O person? Sure, right? Because there's nothing scary on the O, so we're good there. So a B person can take it from an AB person? No, because the A part would trip their system, right? Can they take it from an A person? No. Can they take it from O? Sure, right? Now, what about the AB person? Can they take blood from an A person? Sure, they can, right? Because they've got A2. So their immune system's like, yeah, I know what A is, punted, right? Can they take it from a B person? Yeah, because they got B, right? Can they take it from, of course, they take it from an AB person, right? But can they take it from an O person? Sure, right? So the AB is what we refer to as the universal receiver. No, it can only donate to itself. So AB is a miserable donor because you can't really save anybody other than another AB. Right? So what about O? Can O take A blood? Can it take A blood? No, right? Can it take B blood? Can it take AB blood? Can it take any blood? Other O's, right? But now, can O give it to AB and B and A? Yeah, so O is what we call the universal donor. Right? So we all love the O people because they're going to save the world, right? They can save us all. Yeah, like great. Forced to be a hero when I don't want to be. Darn it, right? By the way, if you're an O-type person, by the way, and you kind of notice when you're just walking down the street that there's like a black van following you sort of suspiciously with blackened out windows. And then you find yourself waking up in a dark alley somewhere with some missing time element associated with your reality and a faint taste of cookies in your mouth. Don't freak out. It's not an alien abduction. It's just Bonfils. They were tailing you. They follow you through the chips that they put in your fillings. They don't do it like dogs where they just inject you anymore, right? They just kind of do it in your fillings. Or if you don't have any fillings, then they get vaccinations, right? So you think you're all getting live virus, you're actually getting microchips by Bonfils, especially if you're O-type. That's the reason why they kind of switch up the vials. If you're O-type, then they'll give you like the microchip in there. Because I mean, nobody wants to follow ABs, right? They're not saving anybody. But that's just, yeah, don't freak out, right? Which is hysterical, right? Because they're doing blood donation today. So they know you're here, O-type people. They know where you are. They're following you. Yeah, which is obviously that's a little extreme, right? I mean, Bonfils, that's kidnapping, by the way, just saying, right? There's any number of laws that are violated, that are a problem with any of those procedures, right? So that's not really a thing, right? The black van's not following you, right? So, well, maybe it is, but not for that reason anyway, right? If there's a black van following you, call the police, because it's not Bonfils. But no, seriously though, I kind of like to play that because when I did that yesterday, it was hysterical. Half of my students had this like look of terror in their face. It's like, you mean the microchips in my feelings? You could see them like hold like they're like freaking. I'm like, guys, relax. I'm just kidding. But actually, O-type people who've donated blood. Actually, I did this once to a student. To the group, and one of my O-type students were like, dude, I know you're kidding, but you're not really that far off. They're serious. I mean, they're like aggressive. So if you're O-type, I mean, they're going to put you on the Christmas list, on the Hanukkah list, on the Kwanzaa list. They're going to make up, you'll fill in the blank, whatever holiday you want, even if it's like a new one, you don't even know what it is. And you want to do that one, they'll put you on that list. I mean, if you don't have any friends, they will come and visit you and they'll bring you some cookies. I mean, they're usually like, dude, they're like seriously aggressive. So with your O-types, yeah, they know that. You guys are loved, right? Bonfils loves you guys. We all do, right? Now the ABs, nobody wants the ABs, right? Because I mean, they can't do anything. That's okay. You know, nobody wants to be my friend for my blood type, so because I'm not an O-type person. So it just must be cool being O-type, being loved by the universe. No, I'm not quite that bad. I'm an A, but nobody cares about A's either. So it's just like it's just like, yeah, Bonfils isn't after me, right? Like, they're not interested. They're like, are you O-type? You go over there then, right? So it's like, oh, they'll tell you. You go walk up to the Bonfils and they'll let you know. They'll be like, hey, line up here, sit down, grab a needle, grab the cookies you want. Okay, so let's talk about cats, shall we? Loves Siamese are cool. I'm not a cat person, first of all, so I freely admit that. I mean, I don't wish any ill will on them or anything like that. Yeah, I'm more of a dog person. I just like the kind of the dumb, lovable thing. Cats are just too master of the universe for me. Enslaving humanity. I mean, seriously, that's exactly what cats are up to. I mean, you know that. And they're obvious about it, right? It's like they're looking at you like, I own you, and you know I own you, and you love it, right? And you're like, yes, I do. Right? It's like, so yeah, I don't. It just kind of, yeah, I can't live with cats. Because I was staying with a friend once, and she had a cat, and I swear I was like just sleeping, and it's like I just had this like dream that there was this cat sitting on my chest, like looking at me, like while I slept, and it wasn't a dream. It was actually happening. She was like, no, that's what he was doing. He was looking at you, like. And then there was like another friend who had a cat, and I was sleeping in their basement, and they had like a red light in there for some reason. I don't know why it was red, but it felt like hell to me. And so it didn't stimulate anything for me. It just kind of felt like, okay, so I just, hell minus the heat. It's kind of what it looked like to me. And I kid you, they had like spiral stairs that came down to the basement, which is cool. But their damn cat would sit right up there on the top of the stairs all night long in a red lighted room, just sitting there, staring at me. I'm all like, okay, so red minus the heat, it's hell. I've got the demon cat staring at me. So I've got the whole thing. All I need is a little fire and we're good. I mean, yeah, so I'm not a cat person. I like a healthy distance between me and cats. So just saying that. Yes. Well, that's because cats own them and they brainwash their owners. They enslave the humanity, right? They basically, and the dogs are here to save us from that, right? That's how it works. They're just a little too dumb, right? Okay, so let's take a look at this. What's going on here, right? So up to this particular point, what we've been talking about are genetic mechanisms, rules, right? Patterns of inheritance by which genes segregate, right? And so it's largely been all genetics. However, when we actually look at this is the first one that kind of is a little bit of an outlier. So it's not an actual mechanism by which genes segregate. This one is more of like a phenotype thing, right? So when you take a look at a particular phenotype, what you're going to see is a combination of your genetics, usually with some sort of pattern of inheritance, like we just explained in the previous list, plus your environment is going to equal your phenotype. Now, generally speaking, if your genetics is very complicated, like for instance, if it's polygenic in nature, and there's a heavy environmental influence that goes into that polygenic trait, then we have lots of variables involved, and we usually call this multifactorial. Multifactorial is a genetic trigger word to say, wow, this is really complicated, right? That's what that means. And it's a tangled mess, and we're not necessarily sure we're ever going to untangle it discreetly, right? A good example of a multifactorial trait is human behavior. Human behaviors that have a clear familial pattern to them suggest that there's a genetic component to it. But obviously, behavior is very slushy. And that bespeaks its environmental component. So we basically put behaviors into multifactorial. This one is a little less tricky than that, but there is an environmental component to it. So we start off with the genetics. Where does the genetics come from? Well, the genetics comes from an enzyme called tyrosinase. Tyrosinase is actually an enzyme that sits at the top of your melanin production pathway. So you take a tyrosine, the amino acid tyrosine, and you convert it into the precursors of melanin. It will eventually become melanin, and you deposit that melanin, your pigment, into wherever you are at that particular point. But it requires active tyrosinase. So where does the genetics come in? The tyrosinase gene is the one that is responsible for making tyrosinase. So there's a gene underneath this enzyme. But here's the problem. In areas of the body where the temperatures are above 33 degrees, and just to give you a reference point, body temperature for us is 37 degrees, right? When it's above 30 degrees, so when it's hot, then tyrosinase is inactive, which means that you're not going to be making pigment. There's no pigment there. And so you're not going to get pigment deposition. So as a result, then you're going to see in the core areas, the hot areas of the body, you're going to see minus pig, right? No pigment laid down because it's too hot. Inactive tyrosinase. However, in areas where the temperatures get below 33 degrees, so these are the cool areas of the body, then tyrosinase becomes active and it starts to make pigment and it deposits that pigment. So that's basically where you see the extremities. Notice where you're seeing this pigment. It's in the areas we associate with cold, right? What are the areas of your body that get the coldest, fastest? It's your hands, your fingers, your extremities, your face, right? So you see extremities, it's the exact same places here that you see the pigment deposition. Now, So this is basically how a Siamese cat basically gets its pigmentation pattern. So you can see you've got, and by the way, it kind of underlies two different ways to control pigmentation, right? There's a genetic control. You can either turn the tyrosinase gene on or off, right? That's a genetic controller. But if it's on, then you have the environmental controller. So you can either have hot or cold, and that's another way to control it. So you can control it on the level of just making the enzyme, first of all, or activating the enzyme, second of all. So you have two different levels of control and regulation that you can utilize in order to control and regulate pigmentation. So, just to give the Siamese a little bit of love, all the cats that are out, I have to admit, if there's a cat that I would like to have just to look at, it would be the Siamese, right? Because like the pigmentations are stunning. Those blue eyes are just like piercing. And it's like, and that's rare in mammals. Blue eyes are not a thing, right? I mean, so to have this kind of look is it's a really handsome looking cat. I mean, that's coming from a non-cat person, right? They will. This is the one, actually, I think it was a Siamese, the one, the devil cat that was looking at me. It was a Siamese. So not only did you have the red, but you had the little blue. So you could see the, yeah. Yeah, so that was creepy. So they're just nice to look at, but they got to stay away from me a little bit. So like, that's the reason I say, I don't want to like have a cat. I just want to look at the cat. That's different, right? So I'm happy looking at it at the, like the pet store and then like leaving it there. Yeah, right. Right. So like I nice. So here's the question I always have, right? It's an obvious question. Think about this for a second. Why would you have a pigmentation pattern like this? But you already are. I mean, pigmentation absorption, it's not really, I mean, it is a thing, right? I mean, dark colors obviously do absorb heat better, but it's not a thing, right? I mean, because our heat regulation is actually more substantial than our pigmentation absorption, right? This question actually came up yesterday in 1111 about this very topic, right? The question was, well, somebody with dark skin, are they going to get hotter because they have darker skin and it's absorbing more energy and it's hotter? Or like animals with dark fur, are they going to be hotter? And the answer is they are absorbing more radiation because that's what dark colors do, but it's insignificant because your thermoregulation in your body is infinitely more robust than the minuscule amount of extra heat you absorb through your pigment. Okay. So you're not hot because you're dark, right? You're hot because it's hot outside. Right. Right. So that's something that's a thing. But why would you have this? So cats are prey, predator animals, right? So let me switch to a prey animal. Let me switch to a Himalayan, not a cat, a Himalayan rabbit, which are white, except the little black muzzle, and they got black ears, and that's it, right? So their idea is like, okay, I'm in the Himalayas, I'm running around in the snow, and then I've got these ridiculously black ears and this black muzzle. So if you think about it, it's the same basic idea, by the way, with this. It's an environmental cue situation. So in the Himalayan rabbit, why in the world would you have black ears and a black face in the middle of a snowbank? Doesn't that seem a little counterintuitive? Doesn't that seem kind of like sticking a big sign on your head saying, eat me? Right? Because every rabbit, if you're a rabbit, you're at the top of everybody's menu, right? So what's the advantage? Why would you do that? Possibly, right? Maybe the ladies find the black ears incredibly irresistible, perhaps, right? It's like, black ears, got it, right? I mean, that may be a thing. That's a possibility. But you're in the middle of a snowbank right now. It's not mating season. Your job is to live, right? Would this be kind of counterproductive to your ability to live? The predator's going to look at you and be like, oh, hey, thank you for having black ears. I picked you right out of the snowbank. So now I'm going to eat you. So did that work out for you, the Himalayan? Why do you do it? It has to work out for you, first of all, because you're doing it. So how does it work? Well, think about it. What a predator sees in a snowbank isn't a Himalayan bunny with black ears. What they see is a little black blob and two black lines moving around. So if you're a predator and you're looking at that and you're like, what is that? Then you're likely going to be more confused than anything, right? Because you're not necessarily sure what you're looking at. But by the way, even if you do know what you're looking at and you go for it, it's like, aha, there's a Himalayan, let's get it. What are you going to attack first? What are you going to grab for? You're going to grab for what you see, right? Which is these black ears. Well, guess what? Did you get the kill shot when you grab the ears? No, because where's the kill shot? In the core, right? The area that doesn't have pigment. That's your kill shot. So what do? You put pigment as a decoy. Because the predator is already confused, is likely not going to see the body, so it won't know how to go for the core. So they'll grab what they see, they'll grab those black ears, but guess what? As a bunny, take an ear. Take it, right? Because I can get away and I can live with half an ear. The masters of this decoy, predatory decoy, we have lots of them around here, actually. No, they're the predators. I'm talking about the prey. Some of you feed them. Some of them eat your car cables. Yeah, the squirrels, right? Okay, raccoons are a different story, yeah, but right. So squirrels are masters of this, right? Because when you take a look at a squirrel, what's the first thing you notice in a squirrel? That big old, obvious, fluffy pom-pom of a tail, which is like as big as the body, right? And then not only that, but when you scare a squirrel, what does it do? It freezes, number one, right? It's like, but then what is it doing with that tail? You see that big old tail in the S shape, right? And it's going like, right, like that. Why is it doing that? Exactly, right? Because the hawk or the fox or whatever who's going to be attacking the squirrel, what's the first thing they see? The tail. So it's like, go ahead and take the tail. Here, take it. Take the tail. Just don't come for the kill shot. Take the tail. Grab the tail. And then, of course, they're like, here, you can have as much tail as you want, right? Look closely at the squirrel population in Denver. There's lots of them that have half a tail. Some of them that have no tail at all. Why? Yep, because that predator avoidance worked beautifully. Okay, that's the exact same thing you're doing with the black ears. So the pigmentation actually is a survival strategy. It's like, listen, if you want to come and kill me, likely what you're going to go for is a foot or a tail or an ear. Have it, right? But you're not going to go for the kill shot. Okay. Oh. Yes, right? Because your core is the most important. You basically stack all of your most critical life core visceral organs in the core, and your body, like as you're going hypothermic, you can actually see your body basically sacrificing your limbs and sort of circling the wagons around your core. Yep, that's exactly the reason why you start to frostbite on your limb, because it's like your body's like, listen, it's obvious that this is not working out well. We're trying to heat up your fingers. We're trying to increase circulation there to get more heat there. But guess what? It's obviously not working, and you're starting to get hypothermic. So what we're going to do is we're going to start retracting from your limbs. We're going to sacrifice those limbs, and we're going to circle the core. Right. So you can live without a few limbs, but you can't live without your core visceral organs. Right. That's exactly what happened. So hypothermia is actually kind of a slow, progressive, just sort of like shrinking down. And eventually, by the time you actually die of hypothermia, I mean, largely your limbs are just ice bricks. It's probably an incredibly uncomfortable way to go. Wouldn't want to. I mean, I don't really want to go eat anyway. They all sound bad, right? Lighting me on fire sounds pretty bad too. Yeah, that's a golfer, right?
Yeah, because I think that's probably the best way to go, because they ran out of pass out.
Yeah, so like, I mean, if you run out of oxygen, you'll just basically black out, essentially what you'll do. Yeah. Well, it does. It does deaden everything, but the problem is it's uncomfortable. I mean, basically what you've got is you're living for a little while with basically blocks of ice. And psychologically, it's not so great because you realize, crap, it's like, this is kind of just, I've got, even if somebody does find me, this is the last time I'm going to see my arm, because we're talking about amputation at this point. So that's how you're looking at it as you're going. Not only that, but it depends on like how you view cold. I mean, for me, cold isn't, I'm a wimp. I don't want to be cold. I don't like it at all. I would rather die of heat exhaustion than hypothermia. So it depends, Like for me, like the most dramatic short story and the most awesome short story ever was To Build a Fire. Read it, Jack London. It's a great one, right? So it's awesome. But that's basically it. The dude ends up dying of hypothermia. It's one of those, you find your body in April kind of things. Okay, epistasis is kind of the last one we have here. This is basically where one gene affects the expression of another. Now I'm going to go ahead and define epistasis with pigmentation because we just talked about pigmentation. And then we're going to take a look at Emerson's data that Emerson found. So in epistasis, basically what you have is, so Emerson kind of did a cross. He did a dihybrid cross, just like Mendel, right? And so he got his F1s that were purple, just like you would expect from Mendel. But then when he got his F2s, instead of seeing 9.331, what he saw was 9.7. That was his ratio for epistasis. That's your diagnostic ratio for epistasis, by the way. If you do a dihybrid cross and the data you get is 9.7, then that's epistasis. Well, where does this come from? It comes from Mendel's basic 9.331 ratio, right? Because if you take 9.331, you have 9. If you add the other 3, you get 9.7. And we'll take a look at that. So here's what I want to do is I want to basically define a system here. So we have a gene for pigment, and the dominant allele will basically make black pigment. The recessive will make brown pigment. But then there's another gene, the E gene. So the dominant will basically make a decision to make pigment. The little e gene will make the alternative decision. Don't make pigment. So in a situation like this, big B, little b, and let's say my E was this, compound heterozygote, what color fur would I make here? Black, right? So what if I had this? Yeah, that would make brown. Now, what if I had this? String. Yeah, right, because what's happening? So in the E gene, it's basically saying don't make pigment. Doesn't matter what B says, because you're not making pigment at all. It doesn't matter if you're going to be making black or brown. So in this case, you're making white. Notice what happens is E is epistatic to B. So the decision in E has to be made first, and that decision permits the expression of B. When one gene requires the permission of another in order to express, that's epistasis. Now, that's the easier version of epistasis. Now, what Emerson saw was a little bit more involved, but we'll follow this here, right? So first of all, you took your true breeders, you crossed them, you got your purple, and then you did your F1 crosses, right? You got your dihybrid cross, right? That's basically what we're seeing here, okay? Now let's take a look at it. So the first thing you saw was a dominant in A, dominant in B, so I got the purple, right? He saw dominant A, dominant B, so he got purple again. Dominant A, dominant B got purple again. Dominant A, dominant B got purple again. Dominant A, dominant B got purple again. Okay, so far so good, okay? If you have a dominant in A and a dominant in B, it looks like it's gonna be purple. Now let's take a look at something that's different. You're gonna have a dominant in A, but now it's gonna be recessive in B, but it's white. So what does that mean? That basically means that what happens in B is actually epistatic to what's happening in A. That is to say that B is affecting the expression of A. In this case, this is saying that B is epistatic to A. Now you have a dominant A, dominant B, you're purple again. Again, B is epistatic to A. Saw that one again. Dominant A, dominant B, dominant A, dominant B. Now you have the opposite, right? So now you have A is recessive, but notice it's white. So that means A is epistatic to B. And again, you see the same thing here. And you have two dominants, and you have B is epistatic to A is epistatic to B, and of course you have both of them together. So what this basically means is that both A and B need to have a dominant allele. They need each other. So what we found out later was that this is actually biochemical pathway, where you have a colorless precursor beginning that is then converted into a colorless intermediate by enzyme A, which is encoded by gene A. And then this colorless intermediate is converted into the purple pigment by enzyme B, which is encoded by gene B. So what happens if you lose B? Well, what happens is A is fine. It'll pile up a bunch of colorless intermediate, but guess what? It's not going anywhere. So you don't get anything, it's white. So that is to say A needs B, right? So B is epistatic to A. So what happens if you lose A? Well, that basically means you're never going to make any intermediate. So it doesn't really matter what B is doing. It doesn't have anything to turn into a purple pigment. So B needs A. A needs B. B needs A. The two of them need each other. So they're acting epistatically on each other. So that is to say the outcome of B is affecting the expression of A and vice versa. Yeah. Oh, the parentals? Yeah. If you change the parentals, that would change everything else. Yeah, because corn can sell. So for those of you table people, this is basically my list, only it's in a table. Okay, so all the patterns of inheritance. So that's the end of 12. And onward to 14. So 14, basically, what we're going to be looking at is DNA. So remember in chapter 10, we talked about cell cycle, right? We talked about G1, S, G2. And we said in S, you're basically replicating your DNA or duplicate and separate. And so the events of that S phase, we said we're going to put on hold. Well, now it's time to talk about it. So this is basically the details, the nuts and bolts of actually what's happening during that S phase. Okay, so it's DNA replication. The first place we have to start, however, is where we left off. Where we left off with Mendel. And so now that we obviously now know that DNA is the genetic material, right? But we didn't know that, right? So when you think about Mendel, remember, Mendel didn't know any of the molecular stuff. He didn't know anything from chapter 10. He didn't know anything from chapter 11. Remember, all Mendel was following was information. That's it. And that's information in these pollen and things like that. We still really didn't know anything about genes or chromosomes or anything like that. So what was the biggest question coming off of Mendel? So now that we have Mendel's work, what was it? Well, very simple, right? Because what Mendel did, remember, the one thing that Mendel did, his conclusion ultimately in the monarchic cross was that the controller for flower color is particulate, right? That there is a particle of inheritance. Well, the question is obviously then what is it? If your results suggest that there's a particle of inheritance, what we now refer to as a gene, then the question is, what exactly is that particle? We didn't know, right? Because remember also after Mendel, we didn't know a lot of things, right? So we didn't really know, like we do now, the macromolecules. We didn't know nucleic acids, carbohydrates, proteins, lipids. We didn't know that very well. We were just starting to discover lipids and carbohydrates and things like that, but we didn't really have a very solid conception in our head that basically this was essentially the macromolecules of life. We didn't have that then. That came much, much later. Now, one of the things that happened, because remember, Mendel was put away for a few decades, and then in about the late 1800s, 1890s to the early century, that's when William Bateson and William Punnett rediscovered Mendel's work, and they started doing Mendel's work in like 4 o'clock plants and corn and Drosophila. And, you know, some people working on their different systems. Some people were working on Drosophila. Some people were working on corn. Some people were working on 4 o'clock plants who do whatever they wanted to work on, whatever their system was. Some people were working on humans. And so Frederick Griffith was one of those. And so one of the things we wanted to understand was, you know, how does all this work, right? So Mendel saw the transfer of information, but the question is, how does this work? And Griffith wanted to sort of recapitulate or recapture that transformation of information. So that's kind of what Griffith was after. And so Griffith's work was happening in about 1928. So just to kind of give you a little bit of historical primer for some context about where the world was in 1928, is we were about 10 years, roughly, not quite 10 years, post World War I. In this era, Germany, under the Weimar Republic, was licking its wounds and paying very dearly the reparations associated with the atrocities of World War I, where many of our Geneva Convention things came out of. And so that was basically happening here. So in Europe, it was a very different world than in the US. In the US here, this was an election year. Calvin Coolidge was ending his second term, and he was done. He had just presided over the Roaring 20s. So this is actually right in the heart of the Roaring 20s. Good times happening here. Everybody's getting rich. Everybody's partying. If you want to read what the Roaring 20s are like, read The Great Gatsby. Yep, exactly. That's where the flappers come from. The zoot suits are coming from here, right? So read The Great Gatsby. That is basically the Roaring 20s culture. That's what's happening here. Of course, we're electing in 1928 our new president, who happens to be Herbert Hoover, who unfortunately gets saddled with one of the worst moments in American history, which happened a year after this, which is what? Yeah, the stock market crash, which happened in 29, and of course the ensuing Great Depression, which we all blame it on Hoover, which really didn't have anything to do with Hoover. The poor guy was just the wrong president at the wrong time. He was just exceptionally unlucky. If it hadn't been for the collapse of the stock market and the Great Depression, we probably have held Hoover in considerably greater esteem. So he was not a bad president. He was just unfortunately ill-timed. Anybody who would have been president at that point would have gotten the same stigma. Yeah. So here's Griffith now. And so what Griffith was doing was studying basically a type of bacteria, Streptococcus pneumoniae. So basically Streptococcus, we know that it's strep throat, right? So Streptococcus is the genus. There's several species in the Streptococcus genus, of which strep throat is a different species. So not the same one here, but pneumoniae is the one that causes pneumonia, right? Now notice in 1928, this is pre-antibiotics, right? Pre-penicillin. And so pre like most medical inventions and technology. So basically if you got pneumonia, it was basically bundle up, go home, get comfortable, kiss the family, start saying your prayers because you're screwed. That was basically it. There was not a lot. I mean, pneumonia now is difficult to deal with. Back in those days, it was lethal. So Griffith had a lot of motivation to try to figure out how to work with pneumonia and ideally how to eliminate it, right? So here's basically what he noticed was that pneumonia, which is caused by bacteria, had 2 different strains. So the Streptococcus strains had 2 strains. One was the S strain, which is virulent, which basically means it causes disease. The other one is the R strain, which does not cause disease. Yeah, so you could be carrying the R strain and it's not going to cause pneumonia. So obviously, Griffith wanted to understand how this worked, right? So what's the deal with this S strain and the R strain and how does this work? How do they interact? What are their roles? And so he wanted to learn more about it. So what he did basically, he worked with mice and he infected mice, hoping to understand a little bit of something about these strains. Because it's in our best interest to understand this, because the more we learn about this, then maybe we may be able to learn some information to beat pneumonia and to save some lives. So here's what he did. So the first thing he did is, he took the S strain, he injected it in his mice, and of course, as expected, he ended up getting dead mice. Right. Then he took the R strain, injected it in the mice, and he got live mice. That looks more like a monkey, but anyway. Then here's the thing we did. Then what he wanted to do was he wanted to treat it. He said, fine, I haven't learned anything new there. I know that the S strain is virulent. I know it causes disease. It's going to kill the mice. I know the R strain is not. It's not going to cause disease. That part I know. But here's the thing. In hospitals, which are basically germ toilets, because that's where we all go when we're sick, right? A good example, by the way, even in today's hospitals, right? Staph infections, right? Most of the time, you never hear about staph infections unless you're in a hospital. That's just because that's where we all are. We're germy. We've got all sorts of bacteria. We leave it there. So hospitals are kind of hotbeds of germ and viral contagion. Okay? That's in today's hospital. Imagine how much worse it would have been in 1928's hospitals. Right. So probably people were actually right to say, like, you know what, I've got a better chance of trying to fight pneumonia at home than going to the hospital, because I could actually go to the hospital and pick something up and then die. And that's probably actually true. Right. Why? Because you just had bacteria all over the place. So one of the natural things you wanted to try out was like, well, wait a minute. What if we killed the bacteria, the bad ones, like the S strains, right? And we use that as a mechanism for sterilization. Think of it this way. It's like when you go to the mountains, you boil your water, right? The idea there is you're killing all the contagion, you're killing the Giardia. So the idea is like, what if we heat treat it? You're going to kill it. And then that way you'd sterilize it that way, right? Well, that's the motivation behind this heat kill. So he said, okay, well, that's fine. We know that the S strain is a problem. What if we kill it with heat? Would that be a viable strategy in hospitals to use in order to sterilize your utensils, right? Kill the S strain with heat and then use it on somebody else. Well, here's the thing. Two things you notice. First of all, there's two things going on here. You can kill the S strain or you can remove the S strain. The two are not the same, right? So if you kill the S strain with heat and you use it on somebody else, you're not removing the S strain. It's still there. Chances are, if you use that scalpel to cut on somebody else, that dead S strain that you killed with heat is going to mix with their R strains, assuming they don't have the S strain because they don't have pneumonia. But those R strains and dead S strains are going to mix together. And the question is, what does that affect? So you're trying to figure out what would happen there. So here's what he did. He took his heat killed S strains, infected his mice. And what did he get? Live mice, because you killed your S strain. But then he killed the S strain, added it to the R strain, and infected the mice. And what did he get? Dead mice. Now, what does this mean? Just these results suggest that it's not good enough to just heat kill it and reuse it. That's not sterile just yet. What you want to do is you have to remove it, right? That's a different thing. So that's kind of helping to inform how hospitals sterilize stuff, especially what Griffiths is doing. So here you can see you basically you're injecting the S strain, you get a little dead mouse here, right? You inject the R strain, you get happy mouse. And when you heat kill it, you get happy mouse, right? And when you add the two, heat killed S strain and R strain, you get dead mouse. And this is basically what he saw. Pretty simple. So what did he see? So what he saw was that in the last group of mice with the heat killed S strain going to the R strain that's alive, he said, there's something in there. There's some inside the S strain cells, there's some sort of a virulence particle that somehow is being transferred from the S strain to the R strain. Why? Because you can see the virulence being transferred from the S strain to the R strain. This process we call transformation. Now, there's two different terms of transformation in biology. There's a cancer one, where you talk about the transformation of a tumor, from like benign to malignant to metastatic. That's the tumor progression. This is not the same thing. This is something a bacteria does, where you basically have transformation, or the transfer of DNA from one bacterial cell to another. And so at this point, you knew that the virulence was being transferred to the R strain, but you didn't know what it was or how that was working. But you knew. So what you did essentially was kind of like a legitimization, if you will, of what Mendel saw, because Mendel also saw the transfer of information. In this case, you're also seeing the transfer of information. It's happening now in these cells, in these pneumonia cells. So you're kind of broadening that story of information transfer. But guess what? You still don't know anything about the particle, do you? So fast forward a couple of decades to Avery, McLeod, McCarty to 1944. So in 1944, basically, we are just coming to the end of World War II. So we've been at it for a few years now. At this particular point, we probably are getting around to, we're starting to tighten the noose on Hitler at this particular point. So the Allies, Marshal Zhukov coming from the east, and the Soviets are coming from the east. We're coming from the west. And so we're starting to sort of strangle Berlin, and we're slowly starting to bring Nazi Germany to. That's happening. So Europe, by the way, is a train wreck at this particular point. London is also, they pretty much had the daylights bombed out of them by Hitler and somehow managed to survive, which was an amazing feat in and of itself. But that's so Europe is just a disaster area at this particular point. Guess what? This is also an election year. So this is actually FDR's last election. So he was elected in 32, and he was reelected and easily in 1944. He died actually a few months after being sworn in of congestive heart failure. As a matter of fact, it's kind of fascinating. FDR was such an enormous personality, and just had such a huge overshadow of American politics and history that we literally elected a dying man. FDR was dying of congestive heart failure in November of 1944 when we reelected him. We knew it. Doctors knew it. But the thing is, FDR was just so immensely magnetic. He saved us from the Depression. He was basically saving us from the war. So he was probably, honestly, to be quite honest with you, I think the wrong Roosevelt is on Rushmore. FDR was unlike anything the U.S. has ever had. The country was completely hypnotized by him. And as it turns out, he was a good guy. So we got lucky. A lot of people think of Obama as being kind of that magnetic personality, that charismatic person. FDR was like 1,000 times more than Obama. And I mean, literally, FDR probably could have done anything he wanted. If he wanted to take the country and become a dictator, we probably would have been like, okay. I mean, he was literally, we literally worshiped FDR as a country. I mean, because that's nobody has ever elected a dying man as president, which was insane. I mean, and it's not like it wasn't obvious. It was obvious if you were paying attention. We just wanted to elect a dying man anyway. I mean, it was just, we loved him so much. And not only that, but it's funny because it's hilarious. Because my grandma was a Democrat who went through that era. We used to tease her because, I mean, she voted for FDR like, 20 years after he was dead. I mean, it's like you'd rather vote for a dead FDR than anybody else, like Adlai Stevenson, for instance. It's like, who? Yeah, that's what we said in 56 as well. But, right, I mean, so it's like, it was just so immense. He was literally the American dictator. By the way, he's also the reason why we have term limits. Yeah, Social Security, pretty much a lot of, Army Corps of Engineers. I mean, all of these things that we live with now, institutions that we have now that we rely on and that we live with, FDR started them. So he was an amazing man. If you ever have a chance to read through the history of that era, the FDR era, read it. It's a fascinating read. But that's what's happening here. FDR is getting elected again, and here comes Abram McClellan-McCarty. Now, here's what we tried to do is we want to basically start making some headway on identifying what this particle is, right? So that's basically what we're going for. So what is this particle of inheritance? That's too loud. So basically, that gets us to Abram McLeod-McCarty. So here's what they did. First of all, what they did is they took cell extracts, which is essentially a nice way of saying cell guts. So they took the cells, right, with this virulence in them, they eviscerated them, right? And then basically everything that is in there that's causing the virulence, including everything that's not, is in this basically the slurry of cell guts, right? So you know that your particle for virulence is in there somewhere. And so the question is, what is it? So then here's what they did. Now, at this particular time, there was two big arguments raging. The two arguments was Team Protein, who said that proteins are the inheritance molecule. And then Team DNA said, no, DNA is the inheritance molecule. And so they're going back and forth. So here's what Abram McLeod-McCarty did. So what they did is they took their cell extracts and then they removed all the protein. And then they asked the question, does it still have the ability to transform cells, right? Does it still have virulence? And what they noticed is that it did not destroy the ability to transform cells. So what's the conclusion from this one? It's not protein. Here's the problem. Here's the pushback. So you removed all the protein. Are you sure? Are you sure you got it all? Okay. Why don't you do an experiment to prove to me that you removed all the protein? Because I'm not going to believe that you actually have the ability, the technical wherewithal to remove all the protein. Think about it. We already know now, they didn't know about chromatin then, but we know now that DNA is wrapped around protein, right? Did they remove the chromatin? No, they didn't. So when they removed all the protein, guess what? They didn't remove all the protein. And that's what the naysayer said. It's like, well, yeah, you might have removed a lot of it, but I don't think you removed all of it, right? So there's a lot of questions there. And none of that, but you're talking about this random slurry of stuff. And most of that stuff, we don't know what's in there. Remember, we only have just a very beginning knowledge of the macromolecules. But we don't really know what's happening in there. We don't really know anything about RNA at this point. This is pre-DNA structure. We don't even know what the structure of DNA is, right? So, I mean, there's a lot, we don't really know what's in there. There's a lot of mystery, mostly question marks associated with this. And so you can see that if you have more questions than answers, you're not going to buy into this. It's like, yeah, is this intriguing? Yes, it is. I'm not willing to sign this check just yet. Okay, so then here's what they said. Okay, fine. So we'll do the reciprocal. So what we'll do now is we'll attack the DNA side of things. So we're going to take some enzymes and we're going to digest up the DNA. The idea is if the DNA is in the inheritance molecule, guess what? You should also digest it, right? So what did they see? They destroyed all the transforming ability. So what's the conclusion from this one? That DNA is the particle. But here's the thing. How well do you understand these digestion molecules? Right? Did you get all the DNA? Is it possible that some of these digestive enzymes that digest DNA are selective? Is it possible that they're not thorough? That they won't digest your DNA completely? Did you get all the DNA? Can you prove to me with another experiment that there's no DNA in there whatsoever? What about other nucleic acids? Did you get everything? What about the RNA? Did you get that? So you can see there's a lot of questions, right? There's more questions really than answers. So in reality, this supports or suggests DNA is genetic material, but it doesn't prove it. It's not convincing enough, right? Because we don't really understand very well what is in that slurry and what all those molecules are, what their roles are. It's going to take a long time to really understand what's going on in there. Okay. So this is just the beginning. So we needed something a little bit more definitive. So here comes the definitive, the Hershey-Chase experiment. So Hershey-Chase basically came along in 1952. So yes, another history lesson. What's happening in 1952? by the way, guess what? It's another election year. For some reason, these guys are working on election years for some reason, I guess. This is the election year actually when Eisenhower gets elected. Truman finished out. He basically took over FDR's term and won again against Dewey in 48. And then he went to 52. At this particular point, what's also happening here is the beginnings of the Marshall Plan, where Europe has been completely bombed out. I mean, completely bombed out because of World War II. I mean, it was devastating. And the Marshall Plan was the U.S. literally was the only country standing in that conflict. And so this is kind of where our foreign policy, our current modern style foreign policy began. You know, our role is sort of like the babysitter for the world. It began here with the Marshall Plan because Europe was on its knees. Japan was completely obliterated. China had just recently was going through the Boxer Rebellion, so they were having issues of their own. The Soviet Union was really intact, but for the most part, it was on us to help Europe rebuild, and that's what was going on in the 50s. That's what's going on in this particular era as well. By the way, if you're keeping track, this is also the heart and core of the baby boomers. So after the war, getting shot at for a good few years or so, we all come home and we start having lots of babies. So from the end of the war, 45 to about 52 is roughly the hotbed of when all those baby boomers were being born. That's the reason why there's a lot of senior living going up right now, because the boomers are getting to their senior years, and there's a ton more of them than there are of us. Go ahead.
I was just going to say for World War II, people think like the U.S. and the Soviets contributed equally, but for every Nazi killed, I think 8 out of every 10 Nazis killed were by the Soviets.
Yeah, no, the Soviets definitely had a upper hand on it, because when we got because we didn't actually get there until D-Day, but our progression was a slow grind because we were trying to liberate France, and that was not easy. I mean, if you take a look at the battles of Bastogne, and I mean, those were grinding battles that were, I mean, brutal. Watch Band of Brothers. That's a great video to watch. You can see what that progression, and we were literally just crawling slowly through France. While on the east, Marshal Zhukov was pretty much just rolling them up. After the mistake of Hitler going after the Soviets, it was easy pickings after that. So they had an infinitely massive upper hand. It's the reason why the Soviets took over the eastern bloc of Europe and of Berlin, is because they got there first and they pretty well dominated the eastern part of Europe in the process. And we dominated the western part of Europe because that's the direction we were coming from, which if you look at the war, it basically everything about the way the world is now and our foreign policy and the way you approach things all was born out of that conflict in World War II. So
another thing too is that when you see like if you can't beat them, just join them. That attitude pretty much in war, any country that loses to America slowly gets Americanized. Like look at Japan now.
yeah, because well, I mean, even if you don't lose to America, I mean, we pretty well Americanize everybody we come in contact with, even if you're not at war with us. That's just the way to, but that's part of the reason why a lot of people will look around, they think, well, how come America is like the dominant country in the world, right? You know, how come? It's because after World War II, we were kind of the only ones left standing. I mean, Stalin was pretty well screwing everything up over in the Soviet Union because he was slaughtering all of his generals because he was whacked out on his gourd. I mean, we were the only functional adult in the room after World War II. This is the reason why the US is like the dominant country now. So that's purely, then that's what it goes back to. Right? FDR. Yeah. Well, that's because he was a bigger man than Stalin, and Stalin knew it. And when Truman came in, Stalin was like, whatever. Yeah, so anyway. Okay, so that's neither here nor there. That was a little poli-sci interlude there. But so what did Hershey Chase do? So basically what Hershey and Chase did was they took bacteriophages, which are essentially viruses of bacteria. And so when you have a bacterium, what a bacteriophage looks like, it's only made of DNA and protein, which really reduces your problem down to just either or. It's very binary in that nature, right? So when you take a look at a bacteriophage, basically the structure of it, you'll have like a little capsule that's made of protein, a little collar piece also made of protein, like these little legs that are also made of protein. And then inside you'll have your DNA genome. And that's it. It's like a little lunar lander, but that's basically what a bacteriophage looks like. So what they do is they come in and they infect the bacterium by landing on them. And then they inject their DNA into the bacterium. At that point, what they do is they hijack the bacteria's processes and they make viral proteins at this point. Instead of your own proteins, they make viral proteins. It's very similar to what the Nazis did, right? When the Nazis roll into town, they take over the baby bottle manufacturing plant. And then they'd say, like, guess what? Stop making baby bottles. You guys are making bullets for us now. So what they did was they infected the baby bottle factory, and then they basically hijacked them and then made them make their own stuff, right? Bullets and stuff like that. They did that throughout Europe. And so that basically, so then you would make all these viral particles, and then you would lyse the cell, and then out comes more virus that can go and infect new cells. This is what's called the lytic cycle. So this pattern of infection, and specifically the hijacking of the actual cell, is the phenotype that we're following. And we want to correlate this phenotype with the genetic material. So here's what we know. We know that the genetic information for this hijack is inside the cell. The question is, which molecule is inside there, protein or DNA, right? At this point, I just told you that we inject DNA, but that came later, right? So at this point with Hershey Chase, we didn't know who gets inside. We do know, however, it's only two possibilities, DNA or protein. And so basically what they did was they differentially labeled DNA and protein, which you can do, right? So you can use the radioactive element of phosphorus, P32, which is radioactive. This basically will highlight DNA, right? So you feed your bacteriophage P32 DNA, and so it'll incorporate that P32 DNA, highlight the DNA only, but not the protein. Then you can use S35, right? Because this is in the cysteines. The sulfhydryl groups, remember that from earlier in the chapter, a functional group, the SH group, which is in cysteines. Most proteins will have a cysteine in it, or 2 or 3 or 4. And so if you feed it radioactive cysteine with S35, then it'll incorporate into the proteins and you'll highlight the protein. Then what they did was they basically tracked radioactive molecules and they asked the question, what's inside with the phenotype? The answer is the genetic material is inside. Well, what is it? The protein or the DNA? And that's basically where they come. So now what we'll do is I'll break this down and we'll talk a little bit about the experiment that they did and how they were able to come to their ultimate conclusion of DNA is a genetic material. And we'll do that at the start of next time.